Dr. Deng, the floor is yours.
Yes, thank you for the introduction. First of all, I would like to thank the organizers for the opportunity to present here today our work, which is about the loss of AMFR as a new cause of autosomal recessive hereditary spastic paraplegia. Human voluntary movement is controlled by the pyramidal motor system, a long CNS pathway comprising corticospinal and lower motor neurons. HSPs are genetically diverse group of inherited neurological disorders, characterized by dysfunction of the corticospinal tracts, resulting in lower limb spasticity and muscle weakness. According to the clinical phenotype, HSPs are classified as pure or complex. The former most common forms manifesting as isolated pyramidal signs, such as spasticity and hyperreflexia, which can be accompanied by sphincter dysfunction and deep sensory loss. The latter presenting with additional neurological and non-neurological features. We encountered two consanguineous siblings who came to medical attention due to developmental delay and mild intellectual disability, which developed progressively into lower limb spasticity. Prior extensive genetic investigations, which included metabolic screening, SNP, HSP gene panel analysis, and trail whole exome sequencing, failed to identify disease-explaining genetic variant, but a shared run-of homozygosity region present in both affected siblings, but absent in the non-affected siblings. Both siblings were included in implementation project for clinical diagnostic whole genome sequencing at Erasmus MC. Whole genome sequencing analysis identified a homozygous protein truncating variant at the first exome of AMFR, which is also located in the shared run-of homozygosity region. Reanalysis of previously generated exomes, that variants were only present 5 reads and were not called by the diagnostic analysis pipeline. In agreement, the average coverage of the mutation site in the 100 in-house WES samples was poor compared to the corresponding WGS samples, thus explaining the previous missing variant in the AMFR. Subsequently, via international collaborators, we identified 5 additional families with in total 15 individuals with biallelic and protein truncating variants in AMFR. All variants were extremely rare and segregated with HSP phenotype in these families. All individuals presented with spastic paraplegia, gait abnormalities, hyperreflexia, and reduced or increased muscle strength. All patients have some signs of developmental delay, mild intellectual disability, or learning problems. On MRI, a thin corpse color zone was found in 5 individuals, but no major dysmorphic features were observed. AMFR is ring finger E3 ubiquitinase anchored on the membrane of endoplasmic reticulum and plays a role in the protosomal degradation of multiple biological processes, which widely expressed, as can be seen in the first and second trimester human fetuses and brain immunohistochemistry. Also, structural protein model of AMFR suggests encountered variants would disrupt most of functional protein domains, including transmembrane and ring domains, which are critical for AMFR functions. To further investigate the disease mechanisms, we generated by CRISPR-Cas9 engineering pluripotent stem cells with an AMFR lockout, which can fully abolish AMFR protein expression, and subsequently differentiated knockout cells into neural stem cells. Subsequent rsync analysis revealed the downregulation genes were enriched for multiple terms related to cholesterol biosynthesis. Earlier study demonstrated AMFR plays a role in the cholesterol mechanism, where the degradation of HMG-CoA reductase. So, in the presence of sterols, insig on the ER membrane bridge AMFR to HMG-CoA reductase, which results in ubiquitination and the subsequent proteasomal degradation of HMG-CoA reductase. Therefore, upon AMFR lockout, HMG-CoA reductase are expected to rise, resulting in more cholesterol synthesis and compensatory downregulation of genes related in cholesterol synthesis, such as sterol regulatory binding protein genes, which we found downregulated in our neural stem cell rsync data. To further investigate this mechanism, we studied neural stem cells using oil red oil staining, which detects lipids. You can see the red dots are lipids in those cells. Whereas the absolute number of lipid droplets did not clearly differ between wild type and AMFR knockout neural stem cells, but the lipid droplets of knockout cells were significantly larger compared to wild types. To confirm, it was directly caused by the absence of AMFR. We performed the rescue experiment, transfecting AMFR knockout neural stem cells using approximate expression wild-type AMFR or approximate expression hypermorphic mutant AMFR. While rescue with wild-type AMFR restored the lipid droplets to the wild-type level, but the restoration of lipid droplets were only partial when rescuing with hypermorphic AMFR. Further demonstrating this lipid droplet phenotype was due to the absence of AMFR. To validate our finding of altered lipid metabolism are also of relevance on patient cells. We next studied patients' fibroblasts and compare those cells with unrelated wild-type cells. We observed the size of lipid droplets was significantly larger compared to wild-type controls. And again, this phenotype could be rescued with overexpression of wild-type AMFR, but only partially when rescue overexpression of hypermorphic mutant AMFR. In similar, using electron microscope, we observed increased lipid droplets and evidence of dilated rough ER in patient fibroblasts compared to wild-type cells, further confirming our findings. Finally, to model the consequences of the absence of AMFR in vivo, we generated an AMFR zebrafish model using CRISPR-Cas9. The mutant zebrafish have normal morphology as the wild types. But oil red oil staining and further quantification showed slightly enhanced overall lipid staining in mutants compared to wild types. Preliminary experiments in zebrafish showed oil red oil staining intensity could be slightly reduced upon the treatment of simvastatin, which is the FDA-approved HMG-CoA reductase inhibitors. It's important to note this requires further confirmation. Also, preliminary locomotor assay showed that showed increase of movements of mutants compared to wild types, which probably reflecting human phenotype. So to summarize, our unpublished work describes a new form of genetically HSP caused by the biallelic protein truncation in AMFR, which we identified in 15 individuals from 6 consanguineous families. AMFR dysfunction results in alteration of lipid metabolism, which can be read out both in knockout neural stem cells as well as patient fibroblasts as enlarged lipid droplets. Also, preliminary experiment in zebrafish suggest this new disorder could potentially be the target for therapeutic intervention, but this requires further confirmations. So as our future plans, we will further investigate lipid droplet size at a subcellular level in zebrafish, like how the lipid droplet size responds to treatment. And also it's very interesting to see whether the setting treatment influences locomotor phenotype, and if so, what kind of developmental stage would the potential treatment have to be applied? Finally, I would like to thank all people involved in this project, in particular the VAHAN lab, diagnostic units of Erasmus MC, our international collaborators, funding agency, and of course, all the members from the Barakat lab, where this project was performed. Thank you very much for your attention. I'm happy to take all questions.
As you are coming to the mic, I have one question from our virtual audience. So from Jasper, you demonstrated quite young HSP patients. Is the AMFR HSP phenotype progressive or quite stable spasticity?
Yes, I think this is this disease is caused by the truncated mutation in the AMFR. I think it would be truncated the whole protein of the AMFR.
Hi, that's great, thanks. I see that lipid metabolism is abnormal in disease, but I'm not sure why you're saying you think that that's what's causing the clinical pathology, and I'm wondering whether there are other targets for AMFR apart from HMG-CoA reductase which might be affected as well.
Yeah, okay. These questions we also talk about in our discussions in our lab. So first, there are some also other HSP phenotypes actually caused by the alteration of lipid droplets or like other cholesterol metabolisms. And because like the human brains also contains the lipid droplets and the cholesterol a lot, and also more and more studies suggest the alteration of lipid droplets or cholesterol metabolisms could cause the brain disorders.
Thank you, nice talk. I observed that 5 of your patients present a severe phenotype, which includes thin corpus callosum as well. So could you relate it with the type of variant you observe in these patients, and are these patients related?
Well, this is a very good question. Okay, your question is really like the specific and related to the clinical phenotype, as I'm a bioinformatician and very focused on the data analysis and the genomic analysis. It's like a tough question for me, but I will definitely come back to you after I have more discussion with our clinicians. Thank you.
And I would have a last question on here. So you showed some movement of the zebrafish model, and so you pointed out that in the dark, the knockout zebrafish are moving less, but in the light it was the opposite. Do you have an explanation or an hypothesis?
This is a great question. I think now we also have some like the very preliminary results, like using the compound heterozygous zebrafish also performed the similar locomotor assay. From the locomotor assay, actually we see like the very reduced movement compared to the white types. I think because in this preliminary results for in the dark, they move less because maybe we just have this, like the small number of those mutant zebrafish compared to the white types.
Thank you.
Yeah, thank you.
So our next speaker is Victor Lopez Soriano is from the University of Ghent in Belgium. He's also a candidate for Early Career Award, and he will speak about an integrated approach using multi-omics data to dissect cis-regulatory role of ultra-conserved non-coding elements in the human retina.